So, biology mechanism of germ cell transformation. Intratubular germ cell neoplasm is thought to derive from malignant transformation of the primordial germ cells or gonocyte during fetal development. So, there is gonocyte or primordial germ cells out of which there is malignant transformation and intratubular germ cell neoplasm is formed. ITGCN. Primordial germ cells. These primordial germ cells migrate from the proximal epiblast yolk cell through the hindgut and mesentery to the genital ridge and become gonocyte. And the precise molecular events underlying mechanism transformation of ITGCN are not well understood. The most consistent genetic finding in germ cell tumor is gain of material from chromosome 12p, and the majority of the NSGCT and seminoma contain 12p, an isochromosome composed of two fused short arms of the chromosome 12. Isochromosome composed of two fused short arms of the 12p means short arm of the chromosome number 12, and the remaining isochromosome 12p negative germ cell tumors also have gain of 12p sequences. If there is 12p negative, then they also have a 12p sequences in the form of tandem duplications and which may be transposed elsewhere in the genome. So gain of 12p sequence has been found in ITGCN, indicating that it is an early event in the testicular cell pathogenesis. It is an early event in the testicular cancer pathogenesis, and this acquisition of this isochrome 12p is not thought to be the initiating event. However, because it is preceded by polyploidization, overexpressed genes of 12p are likely to be important, and there are candidate genes of 12p including several that confer growth advantage like KRAS2, CCND2, cyclin D2 and others that establish or maintain the stem cell phenotype nanoG, DPPA3, GDF3, and the exact genes that are critical to this step has not been identified. So seminomas are usually hypertriploid, whereas NSGCT is more commonly hypotriploid. So seminoma hypertriploid, NSGCT hypotriploid. Other chromosome regions were found to have non-random gains or losses in germ cell tumors that with less frequency of 12p than 12p. Single gene mutation, they are uncommon in germ cell tumor. The KIT ligand pathway has a special relevance of gonadal development. The biologic function of this pathway is broad and include development of hematopoietic cells, melanocytes, germ cells, and KIT ligand is essential for primordial germ cell survival and motility, and are the chemokine SDF1 and its receptor CXCR4, an IHC marker found on primordial germ cells and gonocytes, PLAP, CD117, OCT34, POW-SF5F1, also found on ITGCN, suggesting transformation from these cells during fetal development, and the bioallelic expression of imprinted genes in germ cell tumor has been reported, showing that they are likely arose from primordial germ cells, whereas the genomic imprinting is temporarily erased. And the integrated molecular characterization of testicular germ cell tumor effort published in 2018 analyzed 137 primary testicular germ cell tumor and found a relate paucity of somatic mutations, with only KIT 18%, KRAS 14%, and RAS 4% having significant presence, will all being present in seminoma. And the somatic alterations in KIT found in germ cell tumors are predicted to upregulate pathway activity. KIT ligand plays a role in determining skin pigmentation and has undergone strong selection in European and Asian population, and difference in the frequency of risk alleles of the KIT ligand between European and African population may provide an explanation for the difference in germ cell tumor incidence and between white Americans and African Americans. And there is evidence that epigenetic regulation of gene expression plays a role in pathogenesis of germ cell tumor. DNA methylation patterns, they are different among histological types. Global hypomethylation is more common in seminoma than NSGCT. And in a study of 16 germ cell tumor, the methylation of CpG island in NSGCT was similar to that observed in other tumor types, whereas it was virtually absent in seminoma. Aberrant promoter methylation is generally associated with absent or down-regulated expression of methylated genes, and this can result, for example, in the silencing of tumor suppressor genes. Methylation also has been correlated with the germ cell tumor differentiation, and the more differentiated tumor, yolk sac tumor, choriocarcinoma, teratoma, they were consistently hypermethylated, whereas seminoma and germ cell neoplasm in situ, they were hypomethylated. And some of the observed methylation pattern may reflect normal development rather than germ cell tumor pathogenesis. Now, pluripotency of embryonal-like differentiation in germ cell tumor. Embryonal carcinoma has a 6-gene signature: DNMT3B, DPPA4, GAL, GPC4, POU5F1, and TERF1, which was detected in the 3 of the 5 independent studies, and all 6 of these genes are involved in establishing and maintaining pluripotency. SOX2 encodes a transcription factor essential for maintaining self renewal of embryonal stem cells and is upregulated in embryonal carcinoma. 2 additional genes that encode transcription factors associated with stem cell pluripotency, NanoG and POU5F1, they are upregulated in both seminoma and embryonal carcinoma, and lineage differentiation takes place in NSGCT, mimicking the development of zonal zygocyte, normal zygocyte. Thus, embryonal carcinoma can be thought to as a transformed counterpart of embryonal stem cells, displaying self-renewal, pluripotency, and lineage differentiation. Familial predisposition to germ cell tumor: approximately 1.4% of the newly diagnosed patients with GCT, they have a family history of germ cell tumor, and first degree relative with the patients with testicular cancer have 6 to 10 fold higher risk of development of the testicular cancer, and this observation point to the likely existence of a hereditary germ cell tumor subset. The inheritance effect is mild. The most number of the affected relative in the family is 2, and the age at diagnosis, number of families cases 2 years to 3 years younger than the sporadic, and there is a higher incidence of bilateral tumor. The GR deletion on the chromosome Y, common among patients with the infertility associated with a 2 to 3-fold increased risk of testicular cancer, was studied as a candidate region for hereditary risk. The frequency of GR variant is low, such that it could not only found on a small component of hereditary risk. Genome-wide association studies for testicular cancer have identified total 44 loci of interest, including signal nucleotide polymorphism involved in KIT ligand signaling, regulation of DNA repair, apoptosis, cell termination, male germ cell development, and centrosome cycle. Candidate genes CG, candidate genes GR. Candidate genes repeatedly implicated include KIT ligand, BAC1, SPY-R4, DMRT1. The strongest association of the single nucleotide polymorphism were at 12q22 within the KIT ligand gene, which was there a greater than 2.5-fold increased risk of major allele, and recently a completed meta-analysis of 5 completed this gene-wide association study found 39 single nucleotide polymorphism, including 12 new potential substantiality loci meeting the pre-specified level of significance, and 13 previously described loci that no longer maintain significance. This finding helped define the mutational landscaping up to 37% of the heritable testicular cancer, leaving 63 still unexplained, and strongest association continues to implicate replicate the aforementioned genes, and 8 new candidate loci been found, including one involved in X chromosome. Cisplatin sensitivity and acquired resistance to adult germ cell tumor.